Okay, so it's hard to believe coming here today that it's been a little over 3 years since the IOM report, and I hope with some of the review that you saw presented by Dr. Doroshow yesterday that the members of the IOM committee that gave that important report and members of the community are pleased with both the way NCI went about implementing the report and hopefully with the results of that implementation. Because it's been an intense period, and I think a very productive period for the system. I can't open it. Okay, what did I do with it? Here you go. It is that first one. If not, I can use the other one. Okay, so not to waste too much time, the first slide was simply showing a map of the country with the cooperative group sites, soon to be the NCTN sites, spread broadly across the United States. And the population centers, as you might expect, on the East Coast and on the West Coast are where the heaviest concentration of sites are. But I think an important point that was made by our patient advocate, Mr. Gavin, is there are centers throughout every state in the country, many in the community from our CTEP program, and hopefully bringing the benefits of research to patients in the community, not just patients who have access to major comprehensive cancer centers. Not opening? Okay, thanks. Okay, thank you. So that's the picture, and what I wanted to say here, basically on this slide, is to simply show that in recent years the number of enrollments has come down to around 20,000 per year, and on that distribution up on your right-hand side, about 80% are phase 3 studies, 15% are phase 2, and the remainder are smaller pilot studies. But that number of phase 2 studies, as we discussed a little bit yesterday, is growing, and probably in recent years represents more like 20 or 25% of what the network is now doing. I think the other point that I just wanted to bring home is that to have this big platform across the country is important because this is the one venue where radiation studies are done, surgical studies are done, multiple modality studies are done. These are not just drug studies that are being performed. There's bone marrow transplant studies done through this network, immunotherapy studies. So although I'm going to focus, because of my topic today, on registration trials, often with drugs through the FDA, I wanted to point out that we're talking about cancer control studies, prevention studies, and that's why this platform, even though it's being modified in a positive way, I believe, for the future, is still important to the country. Just quickly go over this. We talk about the annual incidence of cancer in the United States and how many come from trials, patients come from the NCI, and this is just a quick back of the envelope calculation. There's variations year by year in these numbers, but you can see that the NCI system and industry are roughly contributing some 45 to 50,000 patients a year. Other funders may be put on 5,000 patients a year. So, this number probably, if you did this calculation, says that about 6% of patients diagnosed with cancer annually go on trials in the United States, interventional type trials. And that is a far cry from the 23%, I think it was, that we saw in the UK yesterday. Now, we do registration trials in the cooperative group system. There's a long history of this, and some rather important agents have been gone forth to the FDA for approval based on cooperative group collaborations with their industry partners, and I just list them here on the slide. I think it is true that the majority of them are secondary indications, not the primary indication. But secondary indications, of course, can be quite important because often a drug is improved in a advanced metastatic setting, and then we move the drug up to earlier first-line metastatic or adjuvant settings. So these trials actually probably affect the most people with that cancer. And just to go through this quickly for you, occasionally, like on the top one on this slide, the chimeric 14-18 antibody is now coming forward from a COG trial for consideration for an indication in high-risk neuroblastoma, and that would be a primary indication trial. But the majority of the others, the bevacizumab trials in GIST and in first-line renal cell and in non-small cell lung cancer that did receive indications were important studies that were all done with cooperative group support, rituximab, thalidomide, trastuzumab, and then again another study of nelarabine for T-cell ALL and T-cell lymphoblastic lymphoma, which was a primary indication based on cooperative group trials. Now, it's important to note some trials are negative but have important consequences on how we practice medicine. These were potentially registration trials but didn't meet their endpoints. But you can see in dose-dense temozolomide did not work in what was one of the largest brain tumor trials in glioblastoma ever done. Head and neck cancer trials didn't show a role for cetuximab in this setting, or importantly for bevacizumab in adjuvant colon cancer. So the groups have certainly been involved and will continue to be involved in some important trials. One wouldn't want to set up a very large network to do phase 3 trials without these being definitive trials that could change practice. Now, we have done a number of things to try to make sure that the data quality for the NCTN system is quite high and comparable to what's achieved when industry does a study on their own. And I will talk about some of those changes in trial efficiency and quality in terms of the central IRB, some of our CRADA and intellectual property agreements, and some of our new trial designs that we're considering for potential future registration trials. Dr. Doroshow showed this slide yesterday, and I'm not going to belabor it, but I think what's very important is to point out on the bottom of the slide there that we now have this system will function now with adult and pediatric central IRBs. So hopefully that barrier to putting up studies will be overcome. And I think it's rather critical now that we're going to have these smaller molecularly defined populations that institutions have the ability to open trials when they find the right patient, because we probably will have many more trials with small populations. I'll talk a little bit about this common IT data management system. I think that will improve data quality, and we do have this 24/7 patient registration system, which collects a lot of regulatory data in one place, which really does help our industry partners in collaborating with the NCTN system. So the CTSU, the Cancer Trial Support Unit, has been in existence since 2002. It's expanding its services now to cover more phase 2 trials. This is something that most of the cooperative groups requested, and we're trying to support that. And the types of activities are similar to what you'd find in any data center, but what is the power of this is it goes across the entire network. So it really involves itself in tracking reimbursement, tracking the regulatory support, financial management, site auditing, has a help desk, and it can promote and educate physicians and nurses about trials. The IT data management system, this Medidata Rave system that we've introduced in the last year and a half across the entire system, is quite nifty in that we really do have a state-of-the-art system for collecting quality data, as you just heard from our previous presenter, that is critical to being able to go to the FDA and support the needs of our company collaborators. The training costs and overall management costs are reduced because all our groups are utilizing the same system for the first time. And it helps the CRAs at the site in terms of presenting data, because it doesn't really matter which group is leading the trial anymore. And what this will probably do is allow us to meet all the new FDA requirements that are upcoming for electronic data capture, for security, and transfer of the data to them. And from our vantage point as the NCI, it promotes data sharing, because we no longer have very different platforms in our different cooperative groups, so they can actually collaborate on additional scientific projects much more easily and make the data available to other people outside the groups more easily. Toxicity has always been an issue on some of the CTEP-sponsored trials, and I'm happy to report that sitting behind the scenes in Medidata Rave will be a new system to replace what was previously our ADIRS system for collecting adverse events. We now will have a system with a new acronym. It's called CARES. I'm not going to belabor the details of that, but suffice to say it will permit symmetrical reporting of adverse events on all trials, and very importantly, it'll collect adverse events across the entire spectrum from grade 1 to grade 5, and it'll cue the physician if a certain adverse event meets the criteria for a serious adverse event in a particular study. It also, at the FDA's request, will allow verbatim reporting, so it's not only going to be common toxicity criteria grading, but also verbatim when that's indicated. This was gone over yesterday. We needed to be faster. Our industry colleagues said that, time is money. We can't sit around waiting a very long time for NCI-sponsored studies to get up and running. And so we've set some very aggressive deadlines or targets for implementing our trials. For phase 3 trials, we'd like to have them up in 10 months, and 7 months for our phase 1 and 2s. And we did, in addition to having those targets, which are in the red, we have absolute deadlines. And I'll show you that the 450 that's shown on this slide and the 540 have already been reduced down further. So in the interest of time, since Dr. Darashow showed these slides yesterday, I'm just going to go to this last one to show you that, in terms of phase 3 studies, you can already see the rather impressive progress that's been made by the cooperative group system. These deadlines have really worked. Everybody has worked together to really improve, and the top line is basically the bad curve here. This is showing you how much time it took before, and now, since we've instituted these deadlines where nobody is exceeding the deadline and many people are meeting the target, which is the dotted line seen on the slide. So central IRB, I mention this simply because this was often cited as a barrier, and this slide, which was shown previously, makes the point beautifully that if you participate in the central IRB, you can have your trial up and running within 3 weeks, which I think is I think most would agree that that's a great improvement. Intellectual property, of working with collaborators, we need to be able to handle this. CTEP has long had IP agreements for single agent studies and for combination agent studies that have worked pretty well. But then when we came into the era of biomarkers, we had some concerns from many of our pharmaceutical collaborators about how that was going to be handled. And so most of our prior collaborative agreements were pretty silent as to the disposition of agent-treated human tumor samples and the rights related to them from our companies. So we went through a year-long process of seeking input from companies, from diagnostic companies, from investigators, and came up with a revised IP option for our trials. And it's broken into 2 parts. The first part, or this part A, applies to basically the drug itself and any inventions that could come from giving the drug to patients. And here our collaborators receive a royalty-free, non-exclusive license for commercial purposes and a time-limited first option to negotiate an exclusive license for any inventions. And what are we really talking about here? Well, this is if we always cite the minoxidil or Viagra examples. If you were giving a drug and it had another indication other than you intended, that would take care of the IP in those situations. The more important change is this one, and this applies to inventions that come about that are pursuant to clinical or non-clinical studies looking for information, largely biomarkers, from the trials themselves. And here we have a non-exclusive, non-transferable, royalty-free license only for research purposes. Then what we've come up with, and it seems to be acceptable to most of our partners, is a non-exclusive, royalty-free worldwide license to disclose and promote Section B inventions that are necessary or required by a regulatory authority. So simply said, if a diagnostic is discovered on one of our trials, the company who provided the drug and who's our collaborator does not have the right or the first right to that diagnostic. The diagnostic company, the investigator, retains that right. On the other hand, there is no blocking IP so that the company would have to pay royalties every time it wanted its drug used if a regulatory authority said the drug had to be used with that companion diagnostic. And it's sort of a middle of the road position that we've worked on a long time, and most of our collaborators seem to find it satisfactory. So in the interest of time, I'm going to try to tell you about some new trials that we're doing that I think take advantage of this revision that we've made in the cooperative group program. And the first one is called Alchemist. That was mentioned a bit yesterday, and the second one is the Master Protocol in Advanced Lung Cancer. And Alchemist, it stands for Adjuvant Lung Cancer Enrichment Marker Identification and Sequencing Trial. And what it does is it tries to understand that the two new agents that we've waited a long time for in non-small cell lung cancer, those that erlotinib that goes after EGFR mutations and crizotinib that goes after the ALK rearrangement, how do you bring that into the earlier disease setting? And so we're going to have a national trial where we characterize early stage adenocarcinoma of the lung, clinical stages 1 through 3, but they have to be deemed resectable, or they can come in already resected as long as adequate tissue is available. There'll be a test for EGFR and the ALK rearrangement that we're working on identifying whether it will be done locally or centrally. And the patient has to consent to allow donation of their de-identified cancer information for research, central testing of their sample, and 5-year follow-up for treatment and outcome. And in addition, we want to be able to recontact them to consider a rebiopsy if their cancer were to recur. And so the way the tissue will flow on this study is the Alliance will actually set up an umbrella protocol to do screening and follow-up of all the patients, and we're estimating it'll be about 8,000 patients who would need to be screened to find the several hundred that could then be randomized into the ECOG or erlotinib trial or the Alliance crizotinib trial. In the meantime, if they're preoperative, we will be able perhaps to get fresh frozen tissue from some of those patients, but we'll certainly get paraffin-embedded tissue along with germline tissue, a buffy coat from their blood, and a post-op cohort where we'll get formalin-fixed paraffin-embedded tissue. This will all go, the EGFR and ALK testing will be done in a CLIA-approved lab. The rest of the tissue will go off to the Cancer Genome Atlas investigators who will do a series of genomic sequencing, transcriptome, and methylation analyses. And these patients will then be followed either in the trial if they have the mutations or be in follow-up as this data flow trial shows the databases for this trial. And we hope that by having this long-term follow-up, we'll really be able to understand what happens at recurrence to these tumors and what is the mutational status when these tumors, if and when they should recur, which is unfortunately expected in about half of the cohort. This study gives us a lot of opportunities, and some were mentioned yesterday, to do additional research on this large cohort to collect epidemiologic information spanning tobacco, diet, alcohol, work exposure. We're going to learn how to work with these large genome analysis centers, the type that did the TCGA, to do the molecular profiling. We have to develop reports that we can give back to physicians that meet with FDA approval. And we want to develop an approach to sharing unexpected germline mutations for cancer risk with patients screened in this trial, which we're sure to find some and have to deal with that. So these new types of approaches present opportunities beyond just the clinical trial, and we have to take maximal advantage of them. And I think that came through loud and clear in some of the discussion yesterday. Now, another type of trial that we should be able to do, and this was talked about yesterday by Doctors Garraway and Solit, is this basket trial, or what Lisa called the multiple biomarker enrichment design trial. And here we hope to do a phase 2-3 type design where multiple new therapies could be tested simultaneously in a specific disease setting. And we're thinking of non-small cell lung cancer for the setting. We would design this trial to allow FDA approval of new therapeutics. Patients would be assigned to an experimental treatment versus a standard of care control arm on the basis of specific biomarkers. This does have the advantages of the logistic ease that Lisa McShane mentioned this morning. By having them all in one umbrella study, you can slot patients into different phase 2 trials according to their molecular profile. And it adds some consistency to the whole operation and makes better use of the trial infrastructure. We'll have a trial steering committee who can evaluate which drugs and which biomarkers can enter the trial. And we'd like it to really be able to go from the phase 2 level, if we meet our phase 2 endpoints, to a definitive phase 3 confirmatory trial. Each compound's biomarker would be based on an analytically validated test or platform suitable for such a trial. The trial could use a common screening platform for certain biomarkers, but others would need specific and selective platforms. We would have to get probably an investigational device exemption prior to starting the trial, the IDE that was discussed yesterday by Dr. Jessup. And if the new drug shows clinical benefit in selected patient population, then the biomarker could be analyzed and given FDA clearance at the same time as the drug is being evaluated. And lung cancer does have a lot of mutations for which there are targeted agents being developed. And so we would start with a monotherapy approach, looking for these mutations in patients with advanced lung cancer. And then potentially we could even bring combinations into such a trial design. Again, the advantages: it would conserve the samples, easier to standardize, and the sponsors would be able to work with us on the platform and wouldn't have to do this work on their own from scratch. This hasn't been used exactly this way in a registration trial, so we would work closely with the FDA, and we have been doing this in the early days of developing this approach. And basically, just very simply on this slide, patients would come in, they'd have their marker and their drug, and if they hit the phase 2 endpoint of progression-free survival, then we could go on to the phase 3 overall survival endpoint. If they didn't meet the pre-specified endpoint, we would stop that particular arm of the trial. So I think what we've been preparing for, and is now exciting because it actually happened, is we were hoping the NCTN would provide a platform for these kinds of studies. And I think, in fact, the changes that have occurred allow this sort of new science to go forward much easier than it could have in the past. And hopefully we will have the collaborations with our company partners that will enable us to have a group of drugs that could target different mutations, so this type of design would be feasible and practical. Thank you.
Thanks, Jeff. So next up, also from NCI, is Lori Dodd. And Lori's going to talk a little bit about a particularly thorny issue that has come up as we've been using more and more progression events in clinical trials, and that is the necessity of independent radiological review.